Hi. So, in previous session we have initiated the discussion about the antigens, and we have discussed the difference between antigen and immunogen, okay, and we have discussed also about the uh, hapten, okay. So, hapten itself cannot induce the immune response. If you tag this hapten with some carrier molecule, then this can induce the immune response, particularly in terms of antibody production, and this antibody is against the antibody produced is against the hapten and the carrier and the junction between hapten and carrier. Okay. So, in this session let us move on to the properties of antigen, okay, or immunogen, more appropriately properties of immunogens. Okay. So, there are following properties of immunogen that contribute to the immunogenicity. The first is foreignness. Okay. I will explain each property in more detail. So, first is foreignness, second is molecular size, third is chemical composition and complexity, and the fourth is ability to process and present antigen oblique immunogen on MHC. Okay. So, you are aware that once the microbial pathogen is phagocytosed, okay, then this is undergoing degradation, and then after degradation some of these peptide from this antigen, they are presented along with MHC class 2 molecule. Okay. This is for this is a conventional way. Another way is that especially in case of viruses, what is happening, the virus infect the cells, and you know that virus hijack the host cell machinery. Okay. So, after hijacking they start making their own protein. Okay. So, these own protein is basically processed, okay, and then it is presented along with the MHC class 1 molecule to the CD8 T cells, okay, or cytotoxic T cells. Okay. So, this is what I am trying to say in last point, ability to process and present antigen oblique immunogen on MHC. Okay. So, let us discuss about the foreignness. So, what is foreignness? So, if you have an antigen, okay, antigen from same host, okay. Same host means, for example, from any animal, you can use the mice or rabbit, whatever, for doing this simple work. You isolate some protein, okay, for example albumin protein, okay, and then you injected the same protein in the same host. Okay. So, that will be not at all immunogenic, it will not induce the antibody response. Okay. So, in order to have a good antibody response, the protein or the molecule should be from the distinct origin. Okay. So, in a very simple word, the degree of immunogenicity depends on degree of foreignness. For example, I will again explain you. So, if you have a bovine serum albumin is originated from the bovine or cow. Okay. If you take this protein and then inject it in the cow, then it will be not at all immunogenic. Okay. But if you take this protein and inject it in the goat, okay, then it will be a immunogenic. Okay. But if you take this BSA protein and inject it in the chicken, then that will be also immunogenic. However, In case of chicken, it will be highly immunogenic. The BSA which you injected into the chicken is highly immunogenic compared to the BSA which is you injected in the goat. Okay. Why? Because there is a very less degree of difference. Okay. But in case of cow and chicken, there is a great difference. Okay. So, that what I want to explain. So, degree of its immunogenicity depends on the degree of foreignness. So, cow and chicken is more foreign compared to the cow and goat. Okay. So, there are some example. The first example I have already explained. Okay. There are some exceptions also in this theory. Okay. The exception is there are some protein in various hosts. They are highly conserved. Okay, highly conserved. For example, the cytochrome C, the collagen. So, if you take the collagen or cytochrome C from cow and inject it into the chicken, then it will be almost not immunogenic because they are highly conserved. Okay. Similarly, it is true for the collagen, okay, collagen protein. On another hand, in contrast, I will say, because there is a very big difference. Okay. If you take the corneal antigen from the host, for example, you take the mice, you prepare the cornea and prepare the antigen or isolate the protein from the cornea. Okay. And if you inject it into the same animal, for example, in the mice, you have prepared from mice and you will inject it in a mice, then that will be highly immunogenic. Okay. Another is the sperm. Okay. So, if you take the sperm from animal, okay, and then again you will inject it into the same animal, then it will be a highly immunogenic. Okay. These are few exceptions. Now, I will try to explain why it is like that. Okay. So, during development of immunity, okay, at the beginning of development of immunity, at that time these antigen were not exposed, and that is why these antigen is not they do not develop any tolerance against these antigen, the corneal antigen and the sperm antigen or sperm proteins. So, that is why if you prepare this protein and inject it into the host, then that will be a immunogenic. This is one example. Another is if you take out the cell, lyse it, make some DNA or histone or something, and then if you will inject, then that will be also immunogenic. Okay. So, try to understand during education of immunity, these molecules are not exposed, that is why they are not tolerant. Okay. So, this is about the foreignness. Now, let us move to the another part, that is molecular size. So, if the molecular weight of this immunogen will be more than 100 Dalton, this is a generalized concept. If it is more than 100 Dalton, 100,000 Dalton or 100 kilodalton, then it will be a better immunogenic compared to the molecule which is having only 5 kilodalton to 10 kilodalton molecular weight. Okay. So, this is also very important. Okay. If you remember, some of the lipid molecules like derivative of cholesterol, why not we are able to make the uh, uh, antibody against those molecule? There are several regions. One of the region is the molecular size. Okay, I will discuss another region in subsequent slide. Here I am showing some common antigen which we are using in our experiments. Okay, which is quite commonly used in our experiment. Okay, when we perform adaptive immunity experiments. Okay, one is bovine gamma globulin, and its molecular weight is about 150 kilodalton. Okay. Another is BSA. BSA is quite commonly used because it's quite cheap. It is not only used in immunization, it is also used in various other experiment. Okay. So this BSA has a 69 kilodalton molecular weight. Flagellin, it is not so commonly used, but people do use flagellin. Okay, its molecular weight is 40 kilodalton. Hen egg white lysozyme, this is also quite commonly used. This is about 15 kilodalton. Okay, another molecule which is quite commonly used is keyhole limpet hemocyanin. This is a hemocyanin protein. Okay, and its molecular weight is more than 200 kilodalton, as you can see. Oh, sorry, it's a 2000 kilodalton. Okay, ova albumin, this is also quite commonly used in the experiment. Its molecular weight is 44 kilodalton. Sperm whale myoglobulin, this is 17 kilodalton, and this is also used in our experiment. And finally, the tetanus toxoid, okay, which is about 150 kilodaltons. Okay. So these are some molecule which is commonly used in our experiment. Another property is chemical composition and complexity. Okay. What I mean to say that the chemical composition and complexity, here you can see that the molecule which is very simple in structure, they don't have a structural complexity. Okay, as I told you in previous slide that there are the lipid molecule, they do not induce good immune response. One region is molecular weight and another region is this, they do not have any complexity, they are very simple structure. Okay. So you know that a protein molecule has a three major structure, that is primary structure, secondary structure as you can see in this slide, and tertiary structure and quaternary structure. So if the protein molecule or the immunogen or immuno-antigen will have this complex structure, then this will be a very immunogenic. Okay. Here you can I can justify by simple statement. If you make a polymer of same amino acid, okay, then it will be very poor immunogen. Okay. So copolymer of same unit, same basic unit will be not at all immunogenic or almost no immunogenic, either it is amino acid or it is sugar molecule. Okay. Lipids are, as I told you, they do not have a complexity, they do not have a molecular weight, that is why they are heptanes. Okay. They need to conjugate with some complex molecule. For example, BSA, people use BSA, people use KLH as you have seen in the previous slide. Okay. And after chemically joining these lipid molecule with this carrier molecule, they use it as a immunogen. Okay. So I have explained you the importance of this in previous session. Okay. So we make the antibody against these very important molecule, and this these are mainly used in the diagnostics, okay, like a diagnosis for vitamins, some steroid like molecule, as you may know that vitamin D3, okay, it's a cholecalciferol, okay, which is a derivative of steroid, okay. So this is basically the antibody against these molecules are made in this way, okay. Susceptibility to the antigen processing, this is also very important, okay. I have explained you how the antigen is processed in a brief, okay. Bacteria is phagocytosed, then it goes into the endosome, endolysosome or phagolysosome, okay, and over there this bacteria is break down into small pieces, okay, and some of these peptides are presented, okay, the protein molecule, I am talking particularly about the protein molecules, okay. So these protein molecule will turn to the peptides, and these peptides are loaded over the MHC class 2 molecule, and then this will be transported to the membrane, okay, and over there it is presented along with MHC class 2 molecule to the T cells, okay. So now you can understand this thing. So in order to have a better immune response or antibody response, this is very much needed, okay. If you remember, once this antigen is presented along with MHC class 1, then there will MHC class 2 molecule to the TH cells, then these TH cells may differentiate to the TH2, and TH2 immune response is important for the antibody production. So here you can see that the TH2 cell help is needed to make the antibodies, okay. So if this process is hampered, then this will be not very good for development of appropriate adaptive immune response or antibody production, okay. So these two processes, the processing of antigen and presentation is very important in order to get appropriate adaptive immune response, particularly antibody response, okay. So here there is a few points which I would like to say that you can prove it, whether this is important or not. So as you know that human host is having the all protein is composed of L amino acids, okay. It is a polymer of L amino acid, okay. Our all protein has a L amino acid, okay. So what you can do, you can perform a simple experiment. You take a mice or rabbit and challenge this animal with the polymer of the heteropolymer of D amino acid, okay. So when you challenge this animal with D amino acid protein, the protein which is composed of D amino acid, then what will happen? It will be taken up by the cells, but it will be not processed by the host enzyme, because these host enzyme is consist of L amino acid, and they cannot process the D amino acid protein, the protein which is consist of D amino acid. So these proteins will be not immunogenic, okay. On another hand, if you challenge the another group of animal, same composition, same sequence with L amino acid, then you will find out that this will induce very nice immune response in terms of antibody production, okay. So here I think I have explained you in great detail all these things, okay. The processing and presentation of antigen is very much essential, okay. Now, role of biological system in immunogenicity. There are several set roles of the system. Okay, one is that host genetic makeup. Okay, genetics of the host. Okay, I will explain you in more detail. Another is route and amount of challenging antigen or immunogen. Okay, by which route you are introducing this antigen. Okay, and how much amount. If it is extremely low, then it will be not good. Okay, use of some agent, we call it as adjuvant, to enhance the immunogenicity. Okay, now let us take up each one. Okay, the host genetic makeup means genetic makeup of the host. Okay, so you know that there are wide range of polymorphism or changes in the in gene, and when I say gene, then there could be a promoter. Okay, there could be a UTR and so on and so forth. There is a huge things, right? I cannot tell you in few words. Okay, so all these things matters a lot. I am giving you one very simple example for your understanding. For example, there is a MHC molecule, okay, and this MHC molecule is under some promoter. Okay, if there is some changes in the promoter which will express less this MHC molecule, then there will be a not so good production of production of antibody, because the MHC class 1 expression is less, the processed antigen is not presenting very well to the T cells. So this is also coming in the genetics. Okay, like that there are so many genes, right, in B cells, in antigen presenting cells and T cells and so on. Okay, so if anyone if some problem in the gene, okay, in any place which is essential for the expression and all those things, then that will hamper the antibody production, or that may enhance the antibody production. That what I want to say, that genetic makeup is very much important. I have explained you genes of MHC that control the response to the antigen. Another is another example. This is another good example. If there is a mutation in gene segments which is encoding antibody, okay, then that will also hamper the antibody production. Okay. Another example could be a B cell and T cell receptor. If there is some mutation or changes or polymorphism in B cell receptor or T cell receptor, then that will affect the antibody production. Okay. Another is route and amount in which material is presented. Okay. So as I told you, the too low amount of antigen, if it is extremely low amount of antigen, then that will basically induce the tolerance. Okay, too low and too high. So it is very important to find out the correct dosage, okay, of particular antigen or immunogen. Okay. So this is by experience that single dose often not enough. You need a booster dose. Okay, I will tell you in subsequent slide. We give the booster dose. Probably now you are able to understand this concept very well, because you have taken the vaccine for this COVID. Okay, and then you have taken the booster dose. Okay, and now there is a precautionary dose. All these things are because you want to maintain this immune response or antibody response against that antigen. If you will not take this booster dose or precautionary dose, then immune response against SARS-CoV will be decline. Okay, so this is very important to give the booster dose. This depends on antigen to antigen. Okay, for example, if you know that infection of chicken pox or similar infection, smallpox and chicken pox, once you are infected, then you will develop immune protective response throughout your life. You do not infect second time, right? So this depends on the antigen, but in some cases your immune response against that pathogen declines. Okay, and in order to maintain those immune response, you need to take the booster dose. So same thing is true for any antigen and immunogen. Okay, there are various route of immunization, and some route is suitable for some antigen. One is intravenous. Generally this is not so common. Intravenous injection of antigen is very rare. If you give some antigen intravenously, then animal may die. When I say animal, I am talking about the mice. Okay, if you give the intravenous injection of say OVA, then probably that animal will die by shock. Okay, another is intradermal. This is very common. Okay, intradermal, you just place the antigen just beneath the skin. Okay, then the animal will develop appropriate immune response, and there is a possibility that you will see a very good immune response if you challenge the animal through intradermal route or subcutaneous route also. The subcutaneous route is also very common. Intramuscular, you just place the antigen in muscles. In case of SARS-CoV-2, they gave you intramuscular vaccination, particularly I am talking about the Covishield. Okay, intraperitoneal, this is generally we use it in the animal, not in human, but in some cases probably people might be using some antigen or some vaccination may be intraperitoneal, but I am not aware about that. In human it is common or it is an uncommon practice. Okay, another is intranasal. So intranasal is now it is coming up quite common. Okay, so you just introduce the antigen in nose. Okay, and then you can have a immunity against those particular antigen. Okay, another is a use of some agent or adjuvant to enhance the immunogenicity. Okay, so this is also very important. Here you can see that immunogenicity enhances and basically this adjuvant is a immunogenicity enhancing substance. Okay, and there are various mechanisms. So when you will see the composition of these adjuvant, then you will understand that PRR mediated signaling or activation of PRR mediated signaling is very important against any pathogen, and that will give the appropriate bed for the development of adaptive immune response. So adjuvant is basically the PAMs. It's a complex mixture of PAM. I will tell you the mechanism in a short while. Okay, so although mechanism is not very well understood, but it is widely accepted that this adjuvant contains some antigen from or some PAMs from the microbial pathogen. So this is non-specifically activating the innate immune response. This is one aim. Another aim is this makes the antigen phagocytosable. Okay, so for example, if you have a BSA solution, if you inject it, it will be disappear in the host. Either it will cause the shock if the amount is more, or if it is less, it will be immediately cleaved off. So what we do, we make a emulsion. Okay, and you can make a emulsion. Emulsion is basically insoluble kind of condition or state of antigen. Okay, for example, you can see that there is a water in oil adjuvant. Okay, this water in oil adjuvant, we use it in case of animal and particularly in mice. We call it as a Fruent's incomplete adjuvant. It is basically the antigen is present in aqueous form. Okay, and you mix it with the mineral oil. Mineral oil is like a oil kind of thing. Okay, it's a high molecular weight hydrocarbon. Okay, so when you mix it very well, repeatedly or rapidly, then that will make a emulsion. Okay, a globule kind of thing. And when you place this thing in the animal, then this will remain at the site of challenge. Okay, and over there the macrophages and phagocytic cells will migrate and eat up those so they will eat up, and then they will process and present the antigen to the T cells, and then you will have a very nice immune response. Okay, so this is about the Fruent's incomplete adjuvant. There is another thing which we call it as a Fruent's complete adjuvant. So in addition to this mineral oil, there is one more substance is present, which is which provides a PAMs, a wide range of PAMs. Okay, and the substance is heat killed dried Mycobacterium tuberculosis. Okay, or it's a heat killed mycobacteria. Okay, so when you mix this thing in this complete incomplete Fruent's adjuvant, then we call it as a complete Fruent's adjuvant. So here you can see that. So this oil and aqueous thing make a emulsion, and this will be easily phagocytosed by the phagocytic cells or macrophages. In addition, this will also have a PAMs from the mycobacteria. Okay, like lipoarabinomannan, mycolic acid, so on and so forth. Okay, it's a very complex. Okay, and that will non-specifically activate the PRR mediated signaling. So this is a very good situation, and then this will be basically induce a very good antibody response. So now I will talk about some of the adjuvant which is used in animals, and I have told you there is only one adjuvant which is used in case of human. I will come to that point. Okay. So here I have already discussed about the Fruent's incomplete adjuvant. Okay. So basically the mode of action, the probable mode of action for Fruent's incomplete adjuvant is prolonged antigen persistence, and this we achieve by making emulsion, and slowly there will be slow release or slow phagocytosed by the phagocytic cells. Okay. It enhances co-stimulatory signal. Co-stimulatory signal is so of course I have told you the MHC along with antigen is seen by the T cell receptor, but this is not sufficient. There will be some more signal is needed to the T cells from the antigen presenting cells or antigen presenting cells like macrophages and dendritic cells in order to activate the T cells. So those other signals we call it as a co-stimulatory signals. Okay, it induces granuloma formation. So granuloma is just encasing the antigen at particular site, that we call it as a granuloma. It stimulate lymphocyte non-specifically, that you can understand that PRR expressing on various cells including lymphocyte, they are basically stimulated by these PAMs, and then this will make the lymphocytes active. Okay. So Fruent's complete adjuvant is doing this three things. One is prolonged antigen persistence, enhanced co-stimulatory signal, and induced granuloma formation. So another adjuvant is alum. This is aluminum potassium sulfate. This is used in the human. Okay. So of course this is a prolonged the antigen persistence. If you have a good knowledge of chemistry, you can understand. So if you put some salt in protein solution, then that will make a precipitate, right, and this precipitate is concentrated, okay, and when you inject it in the animal, then this will make a small spicules, okay. So this is the principle of most likely the principle of this aluminum potassium sulfate, okay. This also induces the granuloma formation at the site of challenge. There will be a granuloma, okay. People use Mycobacterium tuberculosis, but it is not used in I do not know where it is used. We use the Mycobacterium tuberculosis in case of Fruent's complete adjuvant. Bordetella pertussis, this is also hypothesized to use as a adjuvant. Bacterial LPS, so this is also hypothesized to use it as a adjuvant. However, this is associated with lot of toxicity. So in these cases the lot of toxicity is associated, that is why it is not used. But recently there is a one component of LPS is there, that is MPLA, okay, monophosphoryl lipid A. This component is proposed to used as a adjuvant, and this does not have a toxicity. And there is a synthetic polynucleotide, as I have told you, poly-IC, which is when we introduce in the cell, then it will mimic like a virus infection, but there is no virus, okay. So poly-IC or poly-AU can be also used as a vaccine adjuvant, okay. So with this I am stopping here, and in next session I will discuss a subsequent part of this antigen, and we will also discuss various kind of there is a one very technical term which we call it as a epitope. Epitope is nothing, it is an antigenic determinant. So I will talk about the epitope and all those things in next session. Thank you.